All right, so we'll get started. It's very nice to be here and see a lot of familiar faces and some new faces and get to know everyone. I really like doing these and meeting people and traveling to hear people's stories and what their opportunities have been and what their challenges have been. It helps me learn about what actually I can do better, to what Terry was referring to in making a difference. Terry, Lyle, and the team over here have been really instrumental from this place to be really influential across the country, so I can just bet that. So yeah, you guys need to give yourself a round of applause. This foundation is very important for many reasons. These are my disclosures. I work with a lot of companies, I advise a lot of companies, and I also lead a lot of organizations, so I'm like overly conflicted. So hopefully that means I'm not conflicted. But what I wanted to do is, some of you, I'll keep it informal. If you guys have questions, just raise your hands or like try to get my attention. I'll try to stop and have this like a conversation style. I had trouble and I was always the person in the back of the room like zoning out. So I want you guys to get something out of this today. But there's so much going on in the field that it's been pretty hard to keep up with the information, even for myself. So, and I can only imagine what healthcare teams and ALS specialty teams are trying to do, because there's so much going on in the basic science side, there's so much going on in the clinical research side, and then even the treatments that we have to treat ALS are going through changes, so it's very difficult, especially since January, to really keep up. So what I'm going to do is, first, we'll address the current status. So that's like used to say current treatments for FDA-approved medications for ALS, but I wanted to go over the status because things are changing and evolving and information is coming. So some of this will be familiar to you. A lot of it will be familiar to you, but the nuance is understanding what's transitioning in the field. So one of the players that are here to stay is Riluzole. All of us know this medication. It's been here for a long time. And the reason why I talk about Riluzole at every talk I talk, and if you've seen me online, this slide exists online, so you've seen it. But the nuance of it is that we've had it for 20 years. And why is it still here, and what do we know about it, and why do we still rely on it? So the drug came after 2 clinical trials, right, in the early 90s. And so that approved a dose of 50 milligrams twice a day. It was a small tablet. And then more recently, a liquid formulation and an oral film formulation was developed. But I bring that up not to talk about the drug itself. It kind of made our field ask the question, do we need new formulations, or should we just like kind of look for newer drugs? Why bother, right? Why bother? And a lot of times there were people, physicians, based on the clinical trial say, well, it's a modest benefit, and everyone quotes the 2 to 3 month benefit from the clinical trial. And so we didn't have any other understanding about Riluzole. But there was a study that came out in 2018 around the time when the newer formulations were being developed. And this is a publication from 2018 from the UK group led by Amaral Chalabi. And they applied the King staging system, which was a newer staging system in ALS. It's based on anatomical region, where you're affected. And they applied it to that study, and they found that it prolonged survival and really prolonged it and delayed the time and transition from stage 4 to stage 5. So whenever someone comes to see me, they say, okay, it slows down the disease and prolongs survival. Is that the early part of my disease or the late part of my disease? And there's still some uncertainty about that, but of course, the emphasis in developing any therapies is to preserve independence and preserve that early stage of disease. Now, the hard part about this study is the retrospective study. And if you go back, and there's a lot of new ALS clinicians coming in the field, reading the studies, and we're learning that that study back in the 90s, actually the eligibility criteria allowed people with 5 years of disease or less to enroll. So you could have been further along and enroll in that Riluzole study. So the modest benefit that we saw in that clinical trial might have been bigger if we enrolled people that were earlier, and that's just a hypothesis. But everyone who came into that study was stage 2 or later. So we don't actually know what the benefit is in early stage disease or stage 1. But why do I bring that up? Well, that kind of asks questions like, is it true? Earlier initiation is better. So in 2020, I worked with colleagues, including Dr. Hyman Patterson, to look at real-world evidence of Riluzole. And so in looking at it across population databases, it looked at riluzole, so ever being exposed to riluzole versus never being exposed to riluzole, and the median survival time across those population databases, again, it's a retrospective study, showed a benefit from 6 to 19 months, which is a little bit better than what the riluzole clinical trials showed. So that was in 2020. And subsequent to that, more recently, Dr. Mitsumoto and his colleagues actually published a study looking at the clinical trial database. So this is like a database that houses a whole bunch of information from placebo arms of completed clinical trials. And they were able to kind of look at when riluzole was started and what the survival benefit was. And they could see a statistically significant benefit on survival the earlier it was initiated. So it appeared to reduce the risk of death uniformly, regardless of the time from onset to treatment. But earlier treatment with riluzole may be associated with greater absolute survival from the time of onset. So the reason why I put these slides here is not to talk about riluzole itself, but the way we learned about it as it came on the market and as it was being used, we were able to learn a little bit more depth about the actual treatment more than just what was in the clinical trial. And this is like a foreshadowing to what's coming up next, right? So real-world evidence is really important, and understanding information once something comes to market is really important. So then in 2017, in the US, we get edaravone, also referred to as Radicava. That was based on a 6-month study, placebo-controlled. Everybody was on riluzole as standard of care, and then at the 6-month point, there was a 33% slowing of disease progression as measured by the ALS functional rating scale. Now, what helped support that initial data is that everyone was switched to open label extension, which means everyone got active treatment. And when they were switched to active treatment, it was clear that the folks that were on edaravone from the start in the placebo-controlled portion in the previous slide, in this slide, right? They had a better outcome, and they had a delay to time to onset of events like death and tracheostomy. So again, earlier initiation seemed to have a bigger impact. And so that came in an intravenous formulation in 2017. That posed some challenges for some of us, and I know some of you may have had experiences with it, but you needed an IV access, PICC line, port catheters. That introduced a whole host of other issues for people. And so an oral dose was developed and came to market May 2022. Now, everything was going smooth up until early this year. Another company called Ferrer developed their formulation of edaravone, and they did a study called ADORE. That had a press release that came out in January of 2024 stating that their version of edaravone did not show a benefit on ALS functional rating scale and did not show a benefit on survival. So that created a little whirlwind of discussions and a review, but the details of that study is still not yet public, and that'll come hopefully at the NCALS meeting, which will be June 2024. But there were some key points as you poured through the press release. Number one, the formulation is different. So what we know in the US is a syrup, and you take a syringe and you take 105 milligrams for 14 days as the initial dose, and then you take a 2-week break, and then you take 105 milligrams for 10 days out of 14 days, and then you take a 2-week break. So the dosing schedule was different. The formulation here that was tested were granules and sachets that are dissolved in water. The dose was 100 milligrams as opposed to what we have here in the US, which is 105 milligrams. Different manufacturer, and the clinical trial population was very different. So there are some key differences, but we really can't make any conclusions. So this is something actually, even as physicians, we had some conversations and difference of opinions about how to interpret. So at the moment, there's a consensus that we're going to wait for the information. I certainly have not told people to come off Radicava or edaravone, but we need to learn more before we can make those assumptions. And maybe it's not for everybody, and maybe we will identify certain people that will respond better, but that's how we learn from these clinical trials. So with that said, we were kind of getting our bearings back, and we still had riluzole, we still had edaravone, and of course we had our third drug that came to market in September 2022, which was sodium phenylbutyrate and Terso, which is referred to as Relivrio. That was also approved based on a single phase 2. So normally you have to do 2 clinical trials, or at least a phase 3 and a confirmatory phase 3. But this was a phase 2 study, single study, 6 months, placebo-controlled, and it showed about a 25% slowing of loss of function on the ALS functional rating scale. And again, just like the other study I showed you, when you converted everybody to active treatment in what's called the open label extension, you saw a delay in time to events, or what we call all-cause mortality. And so the red line is the people who were actually randomized earlier and treated earlier with sodium phenylbutyrate and terso. So again, the earlier they were treated, the more impact on all-cause mortality and survival and outcomes. So there's a theme there. So it seemed like we had everything kind of squared away. And then the confirmatory study, so the phase 2 study was approved in September 2022. During the public hearing, FDA said that they would provide regulatory flexibility because of the clinical outcomes that were seen and because there was already a confirmatory phase 3 study called Phoenix underway. Well, the clinical trial report reported out top-line results that showed that it did not show an effect on ALS functional rating scale. And then this is a press release from Amylyx on April 4th saying that they intended to pull Relyvrio from the US and Canadian markets. Now mind you, this press release comes before the actual presentation of the data. So that data was presented April 16th in Denver at the American Academy of Neurology. This is courtesy of Dr. Vandenberg and Dr. Paganoni. They provided the slides for me. And so this study was a 48-week study. So I showed you data from a 6-month, 24-week study, right? So it's a longer study. It was still looking at ALS functional rating scale, but at 48 weeks rather than the phase 2, 24 weeks. And people that were allowed to enter the study could have definite or probable ALS, which is a little bit different than the phase 2 study called Centaur. And so they were randomized 3-2, and then there was an open label extension. Now this is a graph to show you how people landed in the study. Now the phase 2 clinical trial had 137 participants. This had 664 participants, so a much larger study, longer study. The participants, 83% was actually enrolled from Europe, so a smaller percentage from US. The phase 2 study was all enrolled from the US. Why do I make that point? Because there are a little bit of subtle differences in standard of care and accessibility to interventions from region to region. And could that play a part? We don't know. And the other thing, that I'll point out here is there's a category that says other, that although it's basically the same on both arms, that other are the US participants that had to be taken out of the study because it had been approved in the US. So we had a larger than what we normally see discontinuation, a follow-up in a study. Now, this is the primary endpoint, and this is the reason why that press release on April 4th was issued. So the primary endpoint was the change from the ALS functional rating scale, which is a functional scale, from baseline. And so there was no difference at week 48. And they also looked at slow vital capacity and a patient-reported health outcome called ALS AQ40. And so because they didn't see any difference in the primary endpoint, the ALS functional rating scale, or they didn't see any endpoint, they didn't see any difference in the secondary endpoints, they made the decision based on their commitment from the advisory committee meeting in September 2022. But the surprising part at that meeting, so this was the American Academy meeting, is that they did not have survival data yet. So they're still collecting survival data, and so that hasn't even been collected and completed. Biomarker analyses were not completed, and there's still additional data about the population within this study that looked like the phase 2 participants. That's still pending. And so there was a little bit of conversation after the presentation about whether the decision to pull it off the market was a little bit hasty. But, and just to remind you, the database was locked around February of this year, and then they had to make the analyses within 4 weeks, and the press releases all came out within like 6 weeks. That's super fast for a large phase 3 study in any indication. So they worked as quickly as they could, and then there was the pressure to really uphold the commitment made publicly by the company and with the FDA. So we'll see, there's more to come and likely more to be presented in the upcoming meetings. So with that, all hope is not lost, you know. So we, one of the highlights of the past year is tofersen, which is the gene therapy that's approved for SOD1. I'll just put this slide here just to kind of ground us a little bit. This is a whole talk in and of itself, the genetics of ALS, and even now we're getting more into the epigenetics. and risk genes that are associated with ALS. But I put this here because our knowledge of genetic mutations, whether they're risk genes or whether they're pathogenic or disease-causing genes, are growing exponentially each year. And so that means that we are learning more about what's going on in the motor neuron for things for potential targets in the future. But I just highlighted there are multiple clinical trials targeting various gene mutations. And so tofersen is for SOD1, which is about 1% to 2% of individuals, but there's also investigational drugs in late phase clinical trials for C9orf72, for the FUS mutation, and for ataxin-2. And that's just a growing list of gene mutations that have investigational drugs that are being developed. And this is a more recent paper that was published in 2023, and it's just to show you how, when we learn about a new gene, how it actually informs us about what mechanism it may be related to. So understanding its function and understanding that it's associated with ALS can potentially provide us new targets. So if our knowledge of genetics is growing exponentially, that means our potential targets are growing exponentially, and that means that we'll probably get to answers and more meaningful therapy faster. And this is, I just wanted to put tofersen here just for a minute because tofersen is an example of how we're getting there. So ASO, antisense oligonucleotide, is one type of technology that we use for gene-targeted therapy. So antisense oligonucleotides, viral vector delivery, CRISPR gene editing, all the technology is maturing, and it's about us understanding which genes are implicated and how to use or leverage that technology to target those gene mutations. So tofersen is an antisense oligonucleotide. It reduces the synthesis of SOD protein, and there was an international study that evaluated this particular drug in SOD1 associated ALS. And why was it approved? Well, it was approved under accelerated approval pathways. So there's only two pathways in the US that drugs can be approved: accelerated or full approval. Some people use the term conditional, but conditional approval refers to ex-US or sometimes European pathways. So this, what the primary endpoint was looking at ALS functional rating scale at 6 months, but what they saw was that there was a reduction in the concentration of SOD1 protein. So that's what we hoped to do in the first place, right? So it was finally a relief to see that what we wanted to do is what we could see, and we could actually see it objectively in ALS. And then we were able to also see a reduction in the neurofilament in plasma. Neurofilament, a protein that's in your blood when there's structural damage to the nerve. Now, tofersen did not show a statistically significant difference at 6 months, but there was a difference that favored the treatment arm. But that open label extension, so again, you'll see a theme in the clinical trial design, right? I keep talking about 6-month placebo-controlled study, and then I talk about an open label extension. On the open label extension, you saw the difference of the clinical measures being more favorable on the treatment arm than the placebo arm. And so based on the trend on the ALS functional rating scale, the breathing, the muscle measure, and the survival, along with what we saw as biomarkers for neurofilament and for SOD1, it achieved accelerated approval in April of 2023. Now, more recently, there was an announcement from the Committee for Medicinal Products in Europe that they adopted a positive opinion, so it's likely going to receive conditional approval with exceptions in Europe as well, which is positive news for tofersen and for those families affected with SOD1 gene mutations. With all that said, clinical trials still remain important, right, for people living with ALS. We talked about gene therapy, which is really only applicable to about 10% of people, but most people have sporadic ALS, and so we want to get better treatments than what we have right now. So the therapeutic approaches, the STARS actually are areas where we have FDA-approved therapies. So glutamate excitotoxicity is riluzole, mitochondrial dysfunction and ER stress is related to sodium phenylbutyrate and terso, and oxidative stress for edaravone. But you can just see, I can't tell you, if you say, Dr. Andrews, which mechanism is like the most important one in my disease, I can't quite pin it down, right? So we know some of them are more upstream and some of them are more downstream. But this is where the concept of combination, now this is a personal opinion, my personal opinion is that combination therapy is likely what would be ideal and optimal until we have something that targets the disease specifically. And in my visionary kind of ways, I hope that one day that everyone talks about how different ALS is from person to person, right? Someone's ALS could affect their speech, someone else could be arms, someone else could be legs, and the progression could be so different. But ideally, I hope that one day we can say, okay, well, we can test your blood or test something and say that you're predominantly, there's predominant neuroinflammation in you, so we should use anti-inflammatory medications. Or someone might have a lot of glutamate excitotoxicity, so we should use those types of medications. You might have a lot of oxidative stress, we'll use those. But until then, I think combination is the way to go. That was a whole side tangent, sorry. But that's just to show you how we're targeting different areas. And so this, I did a research, so actually this morning I Googled and did a whole search over this slide itself. Takes like hours, like I think 2 days to create and keep up. And so I did the search again. And 2 months ago I did the search and there was 217 studies. Today I did a search and it's 221. Now that's actually filtering down like active or not yet recruiting or recruiting studies only. If you don't put those filters, like 1,000 come up and you can't even make heads or tails of the clinicaltrials.gov database. And the ones that are highlighted in red are the ones that are available or recruiting in the US. The ones that are bolded are ones that are recruiting outside the US. And so what I'll say is baricitinib is a JAK inhibitor, and it's actually being done out of MGH, and it's a cool mechanism that actually affects like FTD and ALS. So they're doing this, what they call a basket study. So you heard a lot about platform studies. The basket is when you have a common, if you have two different diseases, but you have a common marker or an outcome that you can measure, you can enroll those two different diseases, use the same drug, and measure the common outcome or marker. So that's called a basket trial. The platform trial is where you have one disease and one outcome, but you test multiple drugs, right? So you can just tell, I married an Italian guy, so I use my hands a lot. So, and we love charades. So but the concept is innovation in clinical trials. QRL201 is a antisense oligonucleotide targeting stathmin-2. So it essentially, stathmin-2 is a protein that you need for general axonal health. And it's not specific. You don't need to have a stathmin-2 mutation for it. It's actually relevant for any ALS, a sporadic ALS. But what it's trying to do is trying to target a mutation and create full-length normal proteins using the technology we know, antisense oligonucleotide. Dazucorilant is fully enrolled. That's a glucocorticoid receptor antagonist, so again, targeting nerve inflammation. And then the two active compounds in the platform trial is the ABBV is from Calico, and Denali makes DNL363. They both target similar mechanism, EIF2B activator, and what they do is they inhibit integrated stress response. And so if you can inhibit that integrated stress response, you can improve protein synthesis and also hopefully kind of separate out TDP-43 protein clumps. I know there's like physicians and scientists in the room, so like don't judge me for oversimplifying, but that's how I understand it. So, but that's the vision for those EIF2B activators. Ibutalas is in phase 3, and that is a phosphodiesterase inhibitor, and that is something that's to target nerve inflammation. And masitinib is planning a large phase 3 clinical trial also, and that is a tyrosine kinase inhibitor. And then what I will point out is that 2 drugs in the platform trial, CNM-AU8, which is gold nanoparticles from Clean, and pridopidine, which is a sigma-1 receptor modulator, is also moving to phase 3. So there's going to be more phase 3 studies, but what you can see is most of these drugs, this slide is not meant to look for each compound, it's really to show you how we really have a lot of drugs in early phase development and not a lot of drugs in late phase development, so we got to keep the pipeline going. The one of the compounds that was highlighted at the American Academy, and I'm just putting it here, is Prime C. I get a lot of questions about this compound in the clinic. It was presented at the Emerging Science section, and it was the only ALS product that was highlighted at that meeting. Dr. Meritsakovich presented it, and it's a combination, actually a repurposing of 2 drugs that we know about, ciprofloxacin and Celebrex. And so they have a proprietary dosing and proprietary combination, and they did a phase 2 study really for safety and tolerability. but they did show some really good safety. Now, Cipro is an antibiotic. If you take it for a long time, it can cause diarrhea, GI symptoms, even tendon ruptures. So safety is a very important thing to demonstrate with this particular compound. Prime C, you might ask why, Dr. Andrews, why use ciprofloxacin and Celebrex? That seems kind of simple, overly simple. But if you combine them, ciprofloxacin is supposed to target microRNA synthesis, and together with Celebrex, it can affect oxidative stress and also glutamate excitotoxicity. But Celebrex also helps to get ciprofloxacin into the spinal fluid, right? So the blood-brain barrier is a really hard barrier to get past. So that's one of our challenges when we try any of these molecules, especially small molecules that you have to take by mouth, because if you take it by mouth, it gets absorbed in your blood, then it's got to go through your blood and get to your spinal fluid across the blood-brain barrier. So you can just see how challenging it is to develop drugs for ALS. Now, the thing about this was the secondary endpoints was the ALS functional rating scale and SVC, and it showed some trends to favor treatment, but they have biomarker data and neurofilament data that's coming, and they'll likely move to an end of phase 2 meeting with FDA and EMA and move to a large phase 2 or phase 3 study. The other common question I get in clinic is, beyond anything else, I get a lot of questions about stem cells, right? So people ask me, and it's okay. It's very exciting because there's been some basically great information on the preclinical side of how to use stem cells. So I've used the term cell therapy because right now in ALS we're in a special time where we have a lot of early phase products. Some are stem cells, some are T cells. So the terminology, I think in ALS we have to get a little bit better and sophisticated with our terminology, because even with stem cells, they say, oh, I want to do stem cells. Well, where are the cells coming from? How are they treated? How are they manipulated? Are they just the stem cells? So you can get stem cells from your bone marrow, you can get stem cells from your blood, you can get stem cells from your fat cells, you can get stem cells from your skin. And then you can manipulate them to secrete growth factors. You can leave them alone and just put them wherever. So there's a lot of nuance about it. And certainly some of the programs, I think a lot of times when I'm talking to people, they assume that the stem cell clinical trial is trying to replace the motor nerves that are actually lost. But currently the programs are really to provide neuroprotection or reduce inflammation or provide support cells, astrocytes, that kind of help support the motor neuron health. So there's some sophistication, I think, when we talk about stem cells that we have to kind of get used to when we start talking about these programs. So there's T cell therapies, which is different. T cells are involved in nerve inflammation, and if they're not functioning properly, there's increased inflammation, motor nerves are vulnerable, and they die. So there's 2 programs that are using your own own cells to generate healthy T cells. So that's Rapid Therapeutics and Koya. They're actually, you do apheresis, and then the cells go to the company. They make functioning T cells, and then you receive it by IV. There's that's called autologous, your own cell. Then there's allogenic T cell therapy from Cellenkos, where they're using cells not from your body. They're using cells that are derived from umbilical cord blood that's banked, and they create T cells from there with the thought that maybe your own cells will revert back to bad behavior. So if we use cells from other people, they might tend to stay healthy and stay functioning. But the risk there is that when you take other people's cells and put them in your body, you risk really bad immune reactions and life-threatening immune reactions. So we have to be careful with that. Stem cell programs, the most mature program is Brainstorm. Many of you already know about it. There was a press release that confirmed that they worked with the FDA, designed a clinical trial, and they're going to, they actually did what's called a special protocol assessment, and they'll be moving to a confirmatory phase 3 study. There are a lot of reports and stories about people having improvement, and definitely more recently, in April, they published a biomarker paper which showed some promising signals on inflammation and neurofilament, and it's worth looking to see if what we saw in the phase 3 and what people have reported is corroborated in the follow-up clinical trial. Additionally, at the American Academy of Neurology, the Brainstorm study is bone marrow-derived stem cells. This study is adipose-derived, so this is fat cell-derived stem cells. So essentially, there's a fat pad biopsy and you get infusion, both the Brainstorm program and this program, you have to have intrathecal, or where they do a lumbar puncture and infuse the stem cells into the spinal fluid. And so they did that. It was actually safe. You know, the early phase study prior to this phase 2 also showed pretty good safety, but this was really to look at safety and tolerability, which it was, but it was an open label treatment. So unlike the Brainstorm study, which is planned, which is placebo controlled, and a late phase study, this was an early phase study looking just at safety, and was everyone got treated. But there was no strong efficacy signal that they could see, and there was no control arm. There's more data that's going to be presented, but it was clearly safe, and then they're going to try to see if there's any historical controls they can match to see if they can determine efficacy. With all that said, where are we going in the field? And this trial was originally an ALS clinical trial update, and I changed it to the future of ALS, because people ask me, where are we going? You know, and so I wanted to take you, so I just gave you the status, the landscape status. But where are we going? And how are we going to get there? So number one, remember, the start of this lecture, I said, I was talking about the drugs that we have, and I said the earlier we initiate it, the better the effect. So what does that mean? We got to diagnose ALS earlier. And so Dr. Hyman Patterson actually was part of this group that developed a tool for clinicians. So we did launch it at the American Academy of Neurology. I gave it as a lecture. And then I also did just give it to the payer conference for the physicians who do medical reviews on our requests for drugs for ALS. And this is going to be an important tool to diagnose people earlier so we can get therapies and interventions earlier, because it can modify your disease. It can modify your disease significantly. And then the other important chapter or frontier that we have to enter is looking at individuals that are at risk, and gene carriers. Here in Philly, everyone, including the group at Temple, are looking at individuals that are at risk or gene carriers. And this was an old paper from Dr. Benatar, who looked at neurofilament in SOD1. And so what they saw in pre-symptomatic individuals with SOD1 is that 12 months before they actually get their first sign of weakness, so he followed people who had the genes that we know cause ALS, but they didn't have ALS. They had a loved one diagnosed with ALS. And so 12 months prior, there's a spike in neurofilament. And so there's a period where if it goes up in a certain way, we might predict that they're going to convert to ALS. So that might be an opportunity for us to use something to prevent ALS from happening. So what we're doing is taking tofersen and following individuals. So we take their blood every month, and a nurse goes to the person's home, and we follow the neurofilament levels, and when it crosses a threshold, we start treating with tofersen, and the hope is that we prevent ALS from actually happening. So that's kind of where we are with SOD1 ALS, and this, I put it up here because the study is actively recruiting, and if there are people that you know or people that you talk to that want to know whether we can do something, this is open for enrollment globally, actually. And then innovations in clinical trials, I'll just say very quickly, I talked about tofersen, but there are clinical trials actually targeting different mutations. So one product from Biogen, BIB-105, is targeting ataxin-2. The FUS clinical trial is also looking for participants. So the FUS mutation is a tough mutation, because I've been in ALS for almost 20 years, seeing individuals, and we didn't really know about this gene mutation until more recently, but it brings very young individuals, so children, 11, 15-year-old, we had a 20-year-old, and the progression in these young people is fast, less than 1 year. And so it's a very aggressive mutation. It is something that is rare, but we have an antisense oligonucleotide targeting it, and we're hopeful to kind of get that fully enrolled so that we have an answer. So that's a pivotal phase 3 study. And SOD and C9 have several different products that are targeting specific mutations. And the other thing you've heard a lot about is platform trial already. I don't have to talk about the clinical trial design and everything that's involved. This slide is just here to tell you that we started in 2020 in the middle of the COVID pandemic, and by that time, from then to now, we've evaluated 5 drugs, and there's 2 more that are going to be read out at the end of this year. But in doing so, we kind of put away 3, and 2 of them will graduate to phase 3. So the efficiency of testing multiple trials to kind of get through that pipeline that you saw is working efficiently with the platform trial. And then other innovations, we're using technology. Everything ALS is creating a platform, trying to test different remote assessments. ALS TDI is doing it. There's a lot of other groups doing it. But the special thing about this is that they're creating an infrastructure where if there's a new technology that's developed, they can just drop it in here and kind of test it along. And so they've created an infrastructure and a steering committee, and they hold these biomarker summits. I know Dr. Ostro did speak there. Congratulations. And they try to make sure they collaborate with the entire community to really make sure that technology is up to snuff and tested so that we can employ them in clinical trials. And then the last thing I'll talk about is predictive algorithms. So prediction algorithms are algorithms, some people say AI, but this is actually a tool to really use clinical variables and predict how quickly someone's characteristics will change over time. And so there's one by Origin, but there's a couple of other companies here in the US that are developing this tool. There's a group in Europe that is developing the NCAL's risk prediction tool. They're being employed in clinical trials. We don't really know how they're going to behave, so we're testing them now. But how you could use this is, most common right now, it's being used for stratifying clinical trials, meaning, if you ask me, Dr. Andrews, how come we can't get an answer? How come all of the ALS clinical trials are negative? And a lot of people will say, well, that's because ALS is very heterogeneous. And so if a drug is going to work, you got people that look so different, you're not going to see the effect. So sometimes they're slow progressors, sometimes they're fast progressors, and it might be masking your signal. So the stratification means that we're using these prediction tools to predict how people will progress and trying to make sure they're equally allocated in both arms so that we reduce that masking effect. And so that's being employed right now. But I've been to the FDA with Origin to talk about other ways to use it, and so using it as a virtual control arm in early clinical trials, using it to enrich populations so that if you need to look for fast progressors or slow progressors, you can enroll those into a clinical trial. But more recently, we're looking at failed clinical trials and seeing if there's a cluster that actually was a responder group by doing these detectable effector cluster analyses. So you see, like it's a plot, you get a lot of dots, and then there's a cluster that becomes a responder group, and then you'd inform the sponsor that that's the group that they should target for that particular therapy. So that's the cluster analysis method. That's the biostatistics degree talking. So and this is about where we could apply prediction algorithms to in which phase of clinical trials. So you can see across the lifespan of clinical development, you can apply it in every which way, and hopefully that creates some speed and efficiency in clinical development. Last, I'll just talk about some areas that I think are important to kind of focus on in the future for ALS. So one is for sporadic ALS, understanding genetic and environmental interaction, right? So understanding the exposome. So looking at epidemiological studies and understanding what triggers the disease. So even people who are born with the genetic mutation in ALS live for a certain number of time without any symptoms. And then even though they have the gene mutation, so why do they get the disease and what triggers it? And is it that some toxic protein crosses a threshold? Or is it that there's some other factor within their gene code that can influence their expression? Or is it that there's a genetic or environmental interaction that triggers the gene? This might be even true for sporadic ALS, but the step ladder that you see is the number of steps in a model. So for example, for someone with sporadic ALS, there's about 6 or 7 steps before someone would get there. So one of those steps could be an environmental factor, another one could be a physical factor. So this is, I think, the next phase of where we need to study information about ALS. This is actually another exciting area, which is looking at cryptic exons. So in a lot of our basic science meetings, we talk a lot about cryptic exons. I had a hard time understanding this. But the concept is that a TDP43, we hear a lot about that, it causes the loss of function of TDP43 actually causes an erroneous inclusion of these intron sequences, right? These sequences that are not supposed to be carried forward when you make a protein. And they get incorporated into mature mRNA, and that's what's called cryptic exons. And why that's exciting is that for any type of ALS, which is a majority, not all, that are associated with TDP-43 abnormal protein collections, this can be an exciting way to look at a biomarker instead of neurofilament. You've heard about neurofilament. It could also be a potential therapeutic target. And the last is a newer paper that came out. So I talked about antisense oligonucleotide. We've heard about viral vector technology that's going to be launched in ALS. More recently, there was a paper from UCSF that looked at editing gene mutations. So C9 ORF72, which is the most commonly occurring gene, usually it's autosomal dominant. So you have 2 copies of every gene, right? So you have one bad copy of C9 and one good copy. In this particular lab experiment where they looked at cultured human neurons, and so these are neurons that are derived from pluripotent cells that, like skin biopsy, that they turn into little dishes of human neurons. What they saw was that the bad proteins or the products that are developed are generated from that bad gene the mutated copy of the gene. Now, if you take that gene and you cut it out using CRISPR technology, the good gene takes over and all the bad things that happen that are associated with that C9 mutation stop happening. So that means that when you cut out, because the concern is if you take a gene mutation out, by editing it out, do you cause a cascade of events that could be lethal or really devastating to somebody? So they were able to show that cutting it out actually caused the good gene to step up and take an active role and fill in the gaps in a healthy way. So that's like my easy lay interpretation of that graph. So, but why I think it's important, that's a possible strategy to enter into human clinical trials next. So finally, I'll just leave with the fact that there's been a lot of activity in our government that led to the Act for ALS signing in 2021. This has led to the public-private partnership for rare neurodegenerative diseases. So it's not just for ALS, it is applicable to Alzheimer's disease, Parkinson's disease, Huntington's disease, but ALS is the main indication. The FDA was tasked to create an action plan for rare neurodegenerative diseases, and then there's a granting program with additional funds. So a lot of people focus on the expanded access portion, but there is a granting program specific for basic biology and research that's sitting in the orphan products development section of the FDA. Now, last June, FDA did publish their 5-year strategy for improving and extending the lives of people living with rare neurodegenerative diseases. And in that action plan, they specify that they would like to take steps to focus on getting drugs developed faster and providing regulatory flexibility for drugs that are being developed for ALS and neurodegenerative diseases. The NIH also had a strategic planning working group that worked on different aspects, and I think one of the things that they took away from it is actually the optimizing quality of life. I'm biased because I was on that committee with Dr. Jaime Patterson, but that has been a major important planning working group. That document actually gives instructions to the NIH about what they should fund for the future for research. So that's including trying to develop biomarkers, trying to understand the biology, trying to do clinical trials, and also working on ways to fund research that can improve quality of life and quality of care. And last, the National Academies of Science is also working on trying to create a document that will help provide guidance to government agencies beyond the NIH to try to focus on ways that they can make a difference in people living with ALS. And so both the NIH working group and this National Academies ad hoc group have people living with the disease, people at risk, providing input, and that document is going to come out actually next month. And so we're going to wait for that, and that will be a document which I learned is going to be very prescriptive, that gives instructions to Congress about what to create legislation for. It'll give instructions to CMS and Medicare about how they should handle disease, and it also will help provide guidance to the FDA as well. And last, what I'll say is Act for ALS has funded a consortium. It is the largest of its kind that ever in ALS that will create a repository of clinical data and also samples. And What's unique about it is it's ambitious, so its goal is to enroll 2,000 individuals across the country by mid-2026, but it will also include asymptomatic at-risk individuals that are gene carriers as well. So that would be the first time we're trying to do this. And really the thought is that we can grow this all together and try to get all the smaller initiatives across the country kind of all working together as one collaborative group. Because at the end of the day, that's the only way we're going to get an answer for ALS. And so if you ask me, what is the future of ALS, I'll say it's getting pretty bright. And we all have a common mission of having people live better and live longer. And so in order to do that, we've got to do early diagnosis. We've got to get genetic testing out for people as soon as possible and have access to genetic testing for everyone with either family history or even sporadic ALS. Try to get better biomarkers. I really think the environmental exposures is like a major gap in our field. And then also another gap is understanding the information clinically and also biologically of people who are gene carriers that are at risk of developing ALS. And then of course, you heard a lot about innovation in ALS and then promoting open science and collaboration, that's why I'm here. And then also trying to define real world evidence and collecting that so that we can learn from drugs in the future that we'll get. So with that, I'll say thank you for having me and I take any questions.
Does anyone have, yeah, go ahead, Lyle.
Jinsy, wonderful talk, love it. You're absolutely right, there's a lot of excitement about genetic therapies, right? And by the way, one that you didn't mention is at your institution, so Dr. Schneider's Silence ALS program.
So I actually pulled that out, and I'll tell you why.
Yeah.
You finish your question, I'll tell you why.
Well, that's not part of my question, so go ahead. But.
Yeah, I'll just say really quick.
Yeah.
So it is a really exciting program. It is an example, a true example of precision medicine, but it does confuse some people because it is really intended for ultra rare or nano rare. And so I have a lot of people come into my clinic like, I have TDP-43, can I use N-Lorem?
Sure.
And
so that was actually the point I was getting to. So it's an example of this ultra, personalized medicine, right? And you talked about how we're making progress in things like if you have an SOP mutation, we have a drug now, and citing that isn't busting some of these other ones. And the thing of it is that when we do trials for those genetic flavors of ALS, you have what's called a predictive biomarker, and that's the gene test. And you don't enroll somebody in the trial who doesn't have the mutation, right? If we had enrolled everybody with ALS in the reverse trial, it would have freaking failed. There's no way it wouldn't have failed. And yet, we don't do that enough for the trials that aren't genetic therapies. So every medicine that is in a trial for ALS has a biological mechanism. It's something it's targeting. And we absolutely know that those mechanisms have varying degrees of importance in different people living with the disease. And yet, we don't enough, until very recently, have a predictive, so if I'm doing a trial on a particular mechanism, I should make sure the people I'm enrolling are the people who have an abnormality. And it's interesting, one of the trials you mentioned is the baricitinib trial. Right? So that's the one at MGH, basket trial, enrolling Alzheimer's, enrolling ALS, right? That trial is targeting a particular mechanism. And guess what? To get into that trial, they do a spinal tap and they measure a particular thing in your spinal fluid that says, do you have a problem with that pathway? And we're doing that more and more in ALS trials, but that's going to be the answer because it shouldn't be surprising that these trials fail when we're enrolling everybody, right? And yet, so PERV-K trials on certain mitochondrial trafficking, we're doing this more, but I would argue with you that I think that's the answer, less than let's give a multi-drug to everybody. Right? We just need to give the right drugs to the right people because there's, tofersen wouldn't have worked if I gave it to all my ALS patients.
So my brain is breaking it down into 3 sections. So number 1, gene mutations. Obviously, if there's a particular therapy that we can engineer specific for a gene mutation, that actually probably is our biggest bet. That's where we're going to win, right? Number 2, if we can enrich a population. So what you're talking about is enriching a clinical trial population. So if I have a drug and I know that it's going to lower IL-6, then I'm going to screen you for interleukin-6, which is an inflammatory marker, and I'll enroll you in it. And so that's what we were talking about. And it's true, if I know the mechanism by which that drug is going to affect your disease, And so there's two requirements. One, I need to know exactly how that drug works, right? Number two, I need to have an assay that's validated that I can check that biomarker for, and it has to be reliable and acceptable by regulatory authorities for me. And that's one of our Achilles heels in our field. But even with that, I will say we do have a history of doing that. It's just we don't do it often. So the Actemra trial was one where we were looking for people with high IL-6. If they had high IL-6, we enrolled them in the clinical trial. It also means the trial goes slower, by the way. But we knew that was the mechanism of the drug, and we enrolled only people with elevated IL-6 into that study. We also did it for NP001, where they had to have high sensitivity CRP at a certain cutoff, and we enrolled them. So we have done it, not as often as we should. And I think the other pressure there is that we have, as scientists and clinician researchers, a lot of companies want to jump right into ALS, right? And they don't really understand how the drug works, or they don't really know specifically what mechanism it hits in ALS for it to be brought into human clinical trials. So then you can't figure out a biomarker. So it's on us to kind of push the manufacturers as well.
Yeah, I would say we shouldn't tolerate that, right? We shouldn't tolerate these drugs in search of a disease. And I will say the reason I know about the baricitinib trial is because it was funded by the DoD ALS Research Program, which several people in this room have reviewed grants for some people living with the disease. And we require, you can't get that money if you don't have mechanism-specific biomarkers. And I really think it's the only way we're going to get things to work. And so even at the earliest stages of basic science, when we have funding mechanisms from the NIH, from CDC, we need to have those funding mechanisms require that researchers are thinking about that. That's going to be your biomarker.
Then the third group is, say we have everybody and we use the drug on everyone, and we don't screen people for a particular marker. Theoretically, there are two factors that we could do. One is that if I think when we do sporadic clinical trials, we should collect gene mutation information, or even screen for risk genes like UNC13A in a clinical trial. And I think our field is trying to address that, so that if we see an effect, or we can kind of move people back and forth and kind of evaluate whether that's something that masks our treatment effect. The other is looking at, while we're doing a clinical trial, doing exploratory biomarkers within the clinical trial, so that when we finish, although it's still exploratory, we can still segregate patient populations and say, well, it is exploratory, but this cryptic exon showed that these people were better responders. So incorporating exploratory biomarkers, trying to collect gene information, and actually incorporating like UNC13A and ATAXN2 information into them probably will help us in the future. Oh, we got a question? Oh.
Dr. Andrews, thanks. That was a great talk and a great overview. I was just curious, would it be possible for someone to run a clinical trial where they get consent from patients to contribute tissue after their death, and then have those tissues donated to like a post-mortem ALS and cross-reference against Molly Hamel's classification to see if maybe one of these drugs turned out to have a significant benefit for one of the subtypes?
That's a great question. There's someone in this room that can answer that question better than me.
This is what we're doing, so that's right.
Yeah.
Look, everything that we do to validate research in the lab in people living with the disease depends on these unbelievable donations from people living with the disease, of their blood and their spinal fluid and their tissues and their autopsy. We couldn't do anything without.
Yeah.
But I really feel, like you said, we don't always know how a drug works, right? Yeah. I mean, there's a mechanism that a drug is meant to target in the lab. And the fact is, we know from lots of studies that there's clusters of patients, right? That different people have different mechanisms that are dominant. And we've done a really lousy job up until the last couple of years of designing trials to enrich for the right people. So,
the tricky part is it's possible, but it's just that not, like if you do a very large clinical trial, I think one of the challenges is that some centers are really sophisticated and can do that. Like we can consent people, and physicians and researchers will have familiarity with autopsy donations, and we can do that consenting with the clinical trial consenting, but just it's tough because not all centers are capable of doing that or having the logistics of doing it.
It takes money and support staff.
A lot,
Rhizol works, right? That was something we all know works.
Yeah.
Rhizol doesn't lower neurofilaments. It doesn't change it at all.
Yeah.
It's the only drug we have that we're certain it works and slows down the disease, and yet the biomarker that we're most excited about There's no effect.
Yeah, that's a whole nother hour of discussion. Thank you, guys. Any other questions? I got one more.
You were talking about basket trials. Are those usually diseases that are kind of separate, like Alzheimer's and ALS, or more closely related, like ALS and FTD?
Yeah, I think basket trials are probably more appropriate. So it doesn't have to be diseases that are similar in presentation or how they affect somebody, but they should have some similar mechanism. You know, where, like, for example, neuroinflammation is implicated in Alzheimer's disease and ALS and Parkinson's disease. So maybe we can look at PET scan imaging, you know, as the outcome measure for that early phase trial and enroll all three types.
I have a quick question.
Yeah.
I just thank you for your talk. It was interesting and pretty much over my head. But I just can't, like, why is there a gap in trying to figure out why we're getting this disease and the environmental factors? Is it because the funding's not there? Like, why is it so much more funding going into why we're getting ALS?
That's a great question, actually. So you got everything I said then.
Oh,
You
yeah.
totally did. Well, it's funding, right? So funding for that type of research, we have to do, in order to understand environmental exposures that lead to ALS, we have to do large studies, number one. So that's one. Number two, we don't have formal registries. Like, ALS is not a reportable disease, and so, like, we don't have a registry to follow people long term. And so if we caught every case that was a new case that was identified in the United States and followed them and did these questionnaires of, you know, what did you do for a living? Did you have any occupational hazards? What did you eat? How did you exercise? Did you smoke? You know, I think we would get there. But it's a really difficult type of study to do, and has to be done on a large scale, and it requires a lot of money.
And CDC does have epidemiology,
Right.
you know.
CDC, so the thing about the CDC registry, it's helpful that they've done it. They've given us a lot of incidence and prevalence data. Incidence is new cases, prevalence is existing cases, but it's voluntary, right? So if I see 10 patients and only 3 of them decide to put information, we miss out on the 7 patients that could have provided information.
Any other questions before we take a little break?
Thank you.
Thank you, Jinsy.